So the paper I'm presenting today is about the identification of new markers discriminating between myeloid and lymphoid acute leukemia. I found this paper to be easier than the one I first worked on because it didn't go into a lot of the molecular genetics part of it, so it was a bit more simple to understand. And this paper is, I do realize, it's not recent, so the data in it might not be reflective of what's happening today in the research for acute leukemia, but I found it very interesting that they could differentiate between the two based off of specific genes and how they did it. So an introduction to my presentation is that acute myeloid leukemia is a heterogeneous group of malignancies, usually diagnosed based on clinical features, morphology, cytochemistry, immunophenotyping, cytogenic and molecular genetic methods, and recently by differences in gene expression. By saying that it's heterogeneous, it means that two people having the same disease might have different experiences with it. So that's why it's considered a heterogeneous group of malignancies. In this study, to find new markers and create a refined classification of AML, they analyzed two mixed M5 AML patients versus a pool of healthy volunteers by DNA microarray technology. As a result, four potential markers were identified. So their goal was to find specific genes that could help them maybe differentiate between AML patients and the healthy patients, or AML patients and ALL patients. The methods and the material, they started with 38 blood samples from leukemic patients, of which 18 had AML, 3 had chronic leukemia, 8 had BALL, and 9 had TALL. They studied different cell lines and different genes to identify potential genes that could be considered markers for AML. The genes studied covered a wide range of biological pathways, so this way they covered all the different options that they had for the genes. For their results, the microarray analysis showed 63 upregulated genes and 109 downregulated genes in M5 AML patients versus healthy volunteers. So they were able to find a lot of differences in the genes between the healthy and the leukemic patients. They chose of these genes 4 genes, which were MPOCP, AREG, and GNG11, to look further into them and see specifically where they can use them in the diagnosis. So starting off with this image is about the quantification of the expression of selected genes in M5 AML patients. It shows the relative expression of MPOCP, AREG, and GNG11 in 2 M5 AML patients and a pool of 10 healthy individuals. As we can see, the expression of MPO and CP is upregulated in M5 AML patients compared to the control healthy individuals. Meanwhile, the expression of GNG11 and AREG was downregulated, and this is very clear in the pictures. As we can see in the M5, CP and MPO were upregulated, while AREG and GNG11 were downregulated. These genes showed differences between the two M5 AML patients as well. So it wasn't only about the difference between the leukemic and the healthy individuals. They also were different between the two patients that had AML, which is proof of the heterogeneity of AML. The second graph is about the expression of the selected genes in patients with different types of leukemia. So now they looked into the different types of leukemia, and they wanted to see if these genes were able to help them diagnose and differentiate between them. The picture shows the relative expression of MPO, CP, AREG, and GNG11 in AML, BALL, TALL, and CL patients, and a pool of 10 healthy individuals. As we can see in the picture, starting off with MPO, MPO, as expected and as we said in the picture before, is upregulated in AML compared to control. The difference of MPO regulation for TLL and BALL and CL was insignificant compared to control. So therefore, MPO expression can distinguish myeloid from lymphoid leukemia. So they can use this gene to differentiate between the myeloid and the lymphoid types of leukemia. As for CP, CP was upregulated in AML, but it wasn't upregulated in BALL and TALL, and it wasn't really expressed at all in CL. Therefore, CP can be used to distinguish between AML and BALL, and it can also be used to distinguish between AML and TALL. Moving on to AREG, and GNG11, both of them were down-regulated in all of them. They were down-regulated in all types of leukemia, AML, BALL, TALL, and CL, compared to the healthy pool of individuals. But as a conclusion of their research, they discovered that only AREG could be used to distinguish between AML and BALL. As for the third result they got, this shows the expression of the selected genes within patients with different types of AML. So here they took 5 different strains of AML, which were M1, M2, M3, M5, and M6. And they wanted to see how these genes can be used to see the differences between these different strains of AML. CP was upregulated in all of the M1 patients, and the highest CP expression was found in M5. So they concluded that CP can be used to distinguish between M5 and the healthy individuals and the control individuals. Meanwhile, AREG was only significant in M2. To know which is significant and which isn't significant, they looked at p-value and they looked at it from a statistical point of view. And the only one that was actually significant for AREG was the M2. Therefore, they concluded that AREG can be used to distinguish between M2 and the control individuals. And finally, for GNG11, it was downregulated in M1, M2, and M5. So these were the only ones that were significant in the research. So they concluded that it could differentiate between M1 and the control individuals, M2 and the control, and M5 and the control. As a conclusion, to wrap everything together, MPO expression can distinguish between lymphoid and myeloid leukemia. The expression of CP, AREG, and GNG11 has been described for the first time in acute leukemia after this study. GNG11 and AREG are downregulated in AML, BALL, TALL, and CL as compared with healthy controls. AREG can be used to differentiate between AML and BALL, and CP is upregulated in AML and can be potentially used to distinguish between myeloid and lymphoid leukemia. The limitation of this study is that the number of patients that they used wasn't very high, and since the number of patients is limited, further studies were needed with a larger series of patients to actually evaluate the potential of the utility of these genes as molecular markers for AML subtypes classification. So they needed to do further research in the future with a bigger sample size to actually see if they can use these genes or if it was only coincidental with the sample they had. This study is the first to analyze these genes in AML, BALL, and TALL and chronic leukemia patients by RT-PCR. This rapid and sensitive method could be used to screen these genes in different types of leukemia. So this study was actually useful, but to actually have results, they needed to do it on a bigger sample so that they know if they can generalize their results or not. Thank you.